Amino acids are awesome, all of them. And when they join up together through peptide bonds to form a protein, oh. So today I thought let's review what these different amino acids can do, and therefore what the proteins they're incorporated into can do. So over the past month, I've been going day by day through the 20 days of amino acids. I've been going day by day, each day covering one of these different amino acids in its unique R group or side chain that like sticks off out like a charm from a charm bracelet, and therefore it gives the protein that it's incorporated into the properties of that charm. And so these different charms, we can compare them in terms of like their size, their hydrophobicity, their charge, their polarity. We can talk about the metabolism, what we can do with these different amino acids, how we make them, how we break them down. Lots of different things that we can talk about with these amino acids, like both in isolation as well as when they're incorporated into a protein. Because they're typically not acting in isolation, they're acting in the context of a whole protein. And that protein is going to be made up of all these different amino acids. And the different properties of these amino acids are going to influence how that protein folds up to best accommodate each of those unique chains, side chains, each of those unique charms of the charm bracelet. And then how it folds up, you have these different amino acid residues. So basically the leftovers, when they're joined together, we call what was an amino acid now a residue. That's going to be able to give the protein different powers. So if there's like a nucleophile, it can do certain things. If there's charge, it can do certain things. So today I just thought we review these different properties of these individual amino acids, kind of like compare and contrast as opposed to looking at them individually, which I've been doing. And then you can find all the individual posts on my blog and on my YouTube channel, The Bumbling Biochemist. So let's dive in with some compare and contrasting. So the most obvious difference between the amino acids is their size. So we have everything from glycine, which is super duper tiny, and it's got just a hydrogen as its side chain, and then we have tryptophan, which is all huge and big and bulky. And this size matters not just for like beauty purposes, but because it actually influences how the protein can fold and sort of how it can rotate. There's this thing called steric hindrance, which is basically a molecule's way of saying, hey, get out of my space. I need some elbow room. And so in a protein, the rotation around the backbone of the protein is already severely restricted because these peptides are joined through these peptide bonds, that because of the nature of this bond, it has this like double bonded, partial double bond nature due to this like resonance or electron delocalization. Basically, there's only certain places in the backbone that you're able to rotate. So you end up with kind of having this chain of planes that you can rotate only around certain places. And how far you can rotate at those places is going to be restricted by how bulky the side chains are. So if you have something big and bulky sticking out there, it's going to be hard to rotate. Whereas if you have something tiny like glycine, it's going to be super duper easy to rotate. And so glycine is going to be super duper flexible, whereas something like tryptophan is going to be harder to rotate around. And it's not just the like overall mass of it that matters. It's actually how close to the backbone that mass is. So when we talk about things like these branch chain amino acids, where they have this branch point close to the backbone, so like isoleucine and valine, when they have this branch when it's closest to the backbone, it's going to be the harder to rotate around because that bulk is closer to the back, where the rotation is trying to happen, and then you have this like clashing. So if you compare like isoleucine and leucine, although they have the same mass, the leucine has its bulk pushed out farther away, so it's going to be able to rotate more. Now, glycine, it can actually rotate so much that it's not very good at forming these like secondary structures. And so it kind of rotates too much, making it bad for holding things in place, but good if you want to have like loopy regions. Another amino acid that's bad at forming these sort of like secondary structures, these beta strands, and which can pair up into beta sheets, and these like alpha helices. Basically, these are structural motifs, or basically structural forms that often form based on interactions with the backbone. So not with the unique side chains, but actually interactions with the backbone itself, these like hydrogen bonds. These are going to rely on having on the hydrogen from the nitrogen in the backbone. And in the case of proline, the hydrogen is actually tied up because its proline side chain is really weird and it loops up on itself. So not only does it have that bulkiness that's going to make it hard to rotate, but it also doesn't have, because when it's linked into a chain, this hydrogen is going to be isn't going to be there because you're going to have a bond here. And so you're not going to have that backbone hydrogen. You're not going to be able to form those hydrogen bonds. So proline is also not going to be found in these secondary structures typically. Okay, so that was size and kind of flexibility and stuff like that. So remember steric hindrance, atoms saying, I need my elbow room. Get out of my space. If you're bigger, it's going to be harder to rotate. And if your bulk is closer to the backbone, it's going to be harder to rotate. And remember, you need to have that hydrogen available on the backbone in order to form those hydrogen bonds. And so proline is not going to be able to do that. Although size is the most obvious, probably the most consequential and the easiest kind of divider type of thing when you think about amino acid properties is polarity and hydrophobicity, which go kind of side and side. So polarity, basically, that's when you have regions of a molecule that are partly charged. And you have like this like partial separation of charge within a molecule. So all of these different amino acids are made up of different atoms, so of atoms of oxygen and hydrogen and carbon. And basically those atoms are held together by sharing pairs of electrons, and much more on this in other posts. For now, just know that each of these atoms has a set number of protons, which are positively charged, and then they have these electrons, which are negatively charged. If you have an equal number of protons and electrons, you're going to have a neutral molecule. If you have an imbalance, you have what we call an ion, which is the charged molecule. But you can also have a partial, you can have an equal number, but still have partial charges that cancel each other out. And this can happen if you have these atoms not sharing fairly. So they're sharing pairs of electrons to form bonds, but if they don't share them fairly, the electrons are going to hang out more around one of the atoms, say the more electronegative atom that hogs the electrons more. It's going to pull the electrons towards it. And so this is going to make it partly negative and leave the other things partly positive. And so a great example of a polar molecule where you have this separation of charge is water. Oxygen is really electronegative. It's going to pull the shared electrons away from the hydrogens, making the oxygen partly negative, the hydrogens partly positive. And opposites attract. And so the positive parts of the oxygens of one water molecule are going to hang out with the hydrogens of another water molecule. And you get this happening in this giant network of water molecules where the water molecules really like to hang out with one another. They're kind of like sticky. If they will let things into their network, but those things have to offer them something better than another water molecule. They have to offer something else that's some other partial charge or even a full charge. So something that's ionic, something that's charged, or something that's polar, something that has this partial separation of charge is going to be what we call hydrophilic. So it's liked by water, it's loved by water, it loves water. On the other hand, we have things that water doesn't like, these hydrophobic things. These are going to be things without a separation of charge. So these are going to be things that are non-polar, that just have like an equal distribution of the electrons. And often we see these non-polar molecules in something where we have only like hydrocarbons, so hydrogen and carbon, which share electrons pretty fairly. And so these will be non-polar molecules. And so you see these like hydrocarbons, these are just like, there's nothing interesting here for the water to hang out with. So the water is going to exclude them. The water is going to try to maximize its interactions with other water molecules, minimize its interactions with these hydrophobic molecules. And therefore these hydrophobic amino acids are going to be found kind of clustered in the center of proteins. And this is actually going to be a key driving force of protein folding, is the protein like folding up with the water, trying like kind of like cinching around the hydrophobic stuff. And the driving together of those hydrophobic amino acids is going to help the protein fold up. And these non-polar molecules, these hydrophobic molecules, they can still interact. So basically, you can get this like temporary separation of charge just by chance. These electrons are whizzing around, and so we talked about how they're more likely to hang out around a more electronegative atom. And that therefore you can end up with like a permanent dipole where you have that separation of charge like all the time. But because they're just randomly moving around, they might just happen to be hanging out around like a carbon more than a hydrogen in a molecule. Even though those atoms are sharing the electrons pretty fairly on average, if they're randomly, like they just happen to be in one place, well, this can actually set off a chain reaction where you get this separation of charge, kind of this like one happens randomly and then it kind of like kicks off this chain reaction. And then you get these interactions that are collectively strong, but individually weak. So you can get these, sometimes they're called like van der Waals interactions or London dispersion forces, where you get these like temporary random dipoles form that can kind of lead to these effects and lead to things like geckos walking off walls. In terms of the power of something hydrophobic, we talked about how with like sickle cell anemia, you can get mutation in this hemoglobin protein, which carries oxygen through the blood, or you get a glutamate, which is negatively charged when hydrophilic, swapped to a valine, which is nonpolar and hydrophobic. And this can cause the proteins to, as abnormal hemoglobins, to link up and to form long fibers because those hydrophobic this hydrophobic amino acid, which likes to be hidden inside, is now on the surface of a protein. And so it kind of seeks out another hydrophobic niche in another hemoglobin molecule. And this causes these fibers to form that can clog blood vessels and be a real problem. When it comes to these nonpolar amino acids, so these are basically hydrocarbons. So they just have hydrogen and carbon, as opposed to having other more exciting elements, things like oxygen and nitrogen, which are more electronegative. They introduce that polarity, that partial, that charge separation, as well as some other things like nucleophilicity we can talk about. In terms of the hydrocarbons, we just have hydrogens and carbons, which might seem that boring, but they can be laid out in a bunch of different ways. And so we talked about how we could have like we can have branches. So these like branch chain amino acids where you have this branching of this chain. And then you can also have things like straight chains where you would just have a straight chain of amino acids. Those are both examples of aliphatic amino acids. So basically aliphatic is going to be anything other than aromatic. And so when we're talking about aromatic, we're talking about the ones with these rings. And they're not normal rings. So like proline has a ring, but this is not aromatic. What defines the aromaticity is that you have resonance you have this electron delocalization or resonance or conjugation. Basically, you have this sharing of electrons among multiple atoms that's happening in the ring. So basically, in addition to forming a normal bond with their next door neighbor, each of these atoms is also donating some like extra electrons into like a communal pool. And these extra electrons are going to hang out kind of like above and below the plane of these rings. So these rings are planar, they're flat. And then you have this electron, it's like above and below the plane. And much more on this in other posts. But there are basically 4 amino acids that have this resonance. When we talk about aromatic amino acids, we often just like talk about tryptophan, phenylalanine, and tyrosine. Histidine is also aromatic, but we often focus more on its ability to be charged, which we'll talk about in a minute. But as a consequence of this aromaticity, these molecules do some cool stuff. They can interact with like DNA and RNA and stuff because they have this like flat planar thing that can kind of, and DNA bases also have this resonance and RNA bases have this resonance, so they can kind of like form stacking interactions with them, where those like pi clouds are interacting with things. And they can also absorb UV light. And so especially tryptophan, it's going to allow us to measure like concentration of proteins and track proteins with a UV detector because for reasons I talk about in my tryptophan post as well as other posts, basically this resonance is going to lower the cost to excite electrons. And basically it's going to make it easier to absorb UV light and that we can then detect. Okay, so that was the aromatic amino acids. And as I mentioned, histidine isn't often incorporated when we're talking about aromatic amino acids, but we're typically more talking just about these three. The histidine is also aromatic, but what we typically talk about is its ability to be charged or non-charged. So it's classified as one of the what we call basic amino acids. So when we talk about acids and bases, an acid in one definition is something that donates a proton, and a base is going to be something that accepts a proton. And basically, when we talk about amino acids as being basic or acidic, we're talking about in their neutral form. So in their neutral form, histidine, lysine, and arginine are basic. They can accept a proton. And when they accept a proton, they're going to become positively charged. Aspartic acid and glutamic acid are going to be acidic in their basic form, in their neutral form. This means that they can donate a proton. When they donate a proton, they were neutral, now they're negatively charged. And basically, these amino acids can go back and forth between their protonated state and their deprotonated state. And which state they're in is going to depend on the pH, which is a measure of how many protons are around. So if you have a lot of protons around, you're more likely to be protonated. And if there aren't that many protons around, you're less likely to be protonated. pH is an inverse log scale. So this means that the more protons there are, the lower the pH. And the fewer protons there are, the higher the pH. So a high pH is going to be low proton and more basic, more alkaline, whereas the low pH is going to be more acidic and it's going to have a higher proton concentration. And the more protons there are around, the more likely you are to take one. This is going to mean that at a lower pH, you're more likely to be positively charged in the case of these basic amino acids, or neutral in the case of the acidic amino acids. And whether or not the kind of tipping point between being protonated or not protonated, this is going to depend on the properties of the different acids or bases and how much they want that proton. And so if something doesn't want a proton that much, you're going to have to get to really, really high pH levels in order for that or really, really high proton levels, so really low pH, in order for it to get protonated. And this is what we see with aspartic acid and glutamic acid. And so their neutral form is aspartic acid. It has this carboxylic acid group, which can then donate a proton to become its conjugate base, so aspartate. So this goes back and forth between its acid and conjugate base forms. But in our body, it's almost always going to be in this deprotonated form, in this negatively charged form, this aspartate and this glutamate. And how I know this is because if I look at the pKa, I can see that it's really low. Now, the pKa is going to be the pH at which half of it is protonated and half of it is deprotonated. And if I go above the pKa, we're getting to lower proton concentrations, it's less likely to be protonated. And for each difference in pH you go, you're actually going, it's like a tenfold difference. So if you have a pKa of about 4 and our body's about 3, 3 higher than that, you're going to have it be that you're going to have basically 1,000-fold more of it in the deprotonated state. And so you're almost always going to find it in this aspartate and this glutamate form. When it comes to the basic amino acids, well, here, lysine and arginine, they have really, really high pKa's. The pH is going to be way below that. There's going to be lots more protons. And so they're going to typically be protonated. Arginine in particular, because it can form this resonance, this delocalization that we've been talking about in other forms, makes atoms really happy. So the arginine is going to be really, really happy when it's protonated because protonation allows it to share that electrons through resonance. So you're almost always going to find it in this positively charged state. Lysine, you're almost always going to find it in that positively charged state too, but it's easier to pull off a proton. And when you do this, you're going to turn lysine into a strong nucleophile, as we'll talk about. With histidine, it's more complicated. Now, histidine's pKa is actually below the pH, the bodily pH, the physiological pH. So it's predominantly going to be in its neutral form. However, you're also going to get significant amounts of it in the protonated form. And importantly, it can go back and forth between these two. And this allows it to play key roles in like enzyme active sites where it can give and take protons in order to help catalyze or speed up reactions. In their charged forms, these amino acids can do things like form salt bridges, which are basically when you have a positively charged amino acid like lysine or arginine or histidine, kind of hanging out with a glutamate or an aspartate through an ionic bond. So an ionic bond isn't really a real bond. It's not like a covalent bond like we see where the atoms are actually sharing electrons. Instead, you just have something that's positively charged, so a cation, and something that's negatively charged, an anion, hanging out together and being attracted to one another. And so you can get these salt bridges forming. Before I move off of the topic of charge, I just wanted to mention that you might see amino acids drawn in like a weird form that doesn't exist, where you actually have both this non-ionic form, where you have it with both the amino group deprotonated, so in its neutral form, and the carboxylic acid group protonated. And basically, this is not really going to exist because, as we just saw when we saw these similar groups in the side chains, these groups are going to be the nitrogens, these amino groups are going to be in the protonated form, and the carboxylic acid groups are going to be in the deprotonated form in our body. And if you have enough protons around to protonate the nitrogen or to protonate this oxygen, you're going to have way more than you need to protonate the nitrogen. And so what actually exists is going to be this like zwitterionic form where you have the amino group protonated and the carboxylic group deprotonated, carboxylic acid group deprotonated to be carboxylate. And so you're going to see it where you have this like separation of charges, but they cancel out. This is called zwitterion. And so it's not the same as polarity. Well, it's not, I mean, it's type of polarity, but it's not like the same as the partial charge separation. Here, what this finds is it actually has like full charges on these different atoms, but they're going to be spread out. They're going to be separated from one another, and they cancel each other out to give you a neutral molecule. So all the amino acids are going to have like this form. And then what depends if it's actually going to have a charge overall is going to depend on its side chain and whether it has one of those amino acids that are ionizable, so that can take on, that can become charged. And so these are going to be our basic amino acids, histidine, lysine, and arginine, and our acidic amino acids, aspartic acid or aspartate, and glutamic acid and glutamate. So same thing, just different charged forms, these different names. And remember that the acid or base, that refers to the neutral form, but we'll typically go, when we see them charged, that's going to be in their conjugate acid form in the case of the bases, and the conjugate base form in the case of the acids. So that was charge, but we still have a bunch of other things that are polar but uncharged. And these can still do cool stuff, and they can do cool stuff in often acting as nucleophiles. And so basically what makes them have properties that are different than like these nonpolar things is that they have these different like functional groups. So when we talk about functional groups, this is basically anything other than like just like a strict hydrocarbon, just like a strict aliphatic. So we still call like aromatic rings and stuff functional groups, but basically it's anything that can have special properties and do things. You can think of hydrocarbons, the hydrocarbon skeleton, as kind of just being like generic and like filler space, and then if you swap out those hydrogens for other things, you can do cool stuff. And oxygen and nitrogen are going to be and sulfur are going to be nucleophilic. Much more on this in other posts. But basically, a nucleophile is going to be reactive. It's going to be kind of like attacky. It's going to have more electrons than it wants, seek out something with fewer electrons than it wants, and this allows them to kind of form bonds. And I'm not going to go into it here, but you can do really cool stuff. These nucleophilic amino acids are often going to play key roles in enzymes, so there's reaction speeder uppers. So we can see like the serine and serine proteases. It's going to help these proteases cut up proteins. The histidine in RNase A, it's going to help the protein cut up RNA. We see with lysine, we get that in the deprotonated state. Remember, we can deprotonate lysine. We just need a little help. So you might have like an acidic amino acid right next door that could kind of pull off that proton. This makes it deprotonated, makes it nucleophilic, and this is going to allow it to get like acetylated and things. Speaking of which, that acetylation is an example of a post-translational modification. So basically, we have all these different amino acids, and they get incorporated into a protein just like this. When they're incorporated into a protein, that's through a process called translation. So if they get altered after the fact, that would be a post-translational modification. These different amino acids have different like abilities to be post-translationally modified. And so when you have something that has one of these like oxygens or nitrogens, it's going to offer up opportunities for post-translational modification typically. And so like serine and threonine and tyrosine, which have hydroxyl groups, they have OH groups. These can get phosphorylated, histidine too. Cysteine is going to be a special case I'll talk about in a second. Lysine can get acetylated and can get methylated. Proline can get hydroxylated, so lots of cool stuff. So I said I'd talk about cysteine in a minute, and why I said this was because cysteine is one of the two amino acids that has a sulfur. And so basically in a cysteine, you have this silyl group, this SH group, which can deprotonate to form a thiolate, or it can get oxidized. And when it gets oxidized, it can actually form these like cross links. And so these are actually going to be covalent bonds between like different regions of the protein or between different proteins. These are going to be like real bonds, unlike those like ionic bonds. They're actually sharing electrons. But sulfur, for reasons I talk a lot more about in my posts on cysteine and methionine, which is the other amino acid that has a sulfur, sulfur basically it forms weaker bonds. It's got kind of like a bigger electron cloud. It's harder to form strong bonds, and so it's going to be easier to break those up. And so you can kind of get these temporary permanent bonds, if that makes sense. They're covalent bonds, but they're easier to break. And so these crosslinks are why you add your like reducing agent like DTT or BME to your like buffers and to that sort of thing in the lab. But basically, yeah, so cysteine can form those crosslinks. Methionine doesn't do the cross-linking, but it does allow you to kind of take this methyl group on the end of methionine and transfer it to other things through an S-adenosine methionine intermediate. And so much more on that methylation in my post on methionine. Okay, so some last things. Let's talk about how we can make and break these. And when we're talking about making and breaking molecules, we're talking about metabolism. So metabolism encompasses anabolism, which is molecule making. Think of like anabolic steroids, and catabolism, which is molecule breaking. Think of your cat like tearing up your couch. Often catabolism is breaking down molecules for energy or for smaller reusable parts. There are many, many interconnected pathways that can go either way. So metabolism is more like a complex subway system than like nice linear pathways. It's like more like webby. And so molecules can be interconverted and transferred and made into different forms, whether you want to make different molecules with them. Basically, because they're all made up of the same sort of atoms, you're able to kind of like interconvert between different things. So some of these amino acids are what we call essential, and this is in the dietary sense, meaning we need to get them through our diet. We can't make them ourselves, at least not like with a net gain. So some of these we could make, but it's just not like energetically efficient. And you don't get like a net gain of that amino acid. Whereas others you're able to easily make yourself. And so the essential would be dietary essential. You need to make them yourself, or you need to get them through your diet. Things like phenylalanine. Okay, and then you can also convert amino acids, use them for different fuel sources. Often when we're talking about these different conversions, we're dealing with through some intermediate of the tricarboxylic acid cycle, which is like a central hub of metabolism. You can get this like acetyl-CoA from like the glycolysis, so from the breakdown of sugar. And you can get other intermediates from other things like the breakdown of lipids. When you break down lipids, you can get these things called like ketone bodies. You can also get these ketone bodies when you break down certain amino acids. Now, these ketone bodies are they can be used for a couple different things. They're a way that your cells can like transport like fat energy type stuff through the bloodstream, because the fats aren't going to be water soluble, but these are, so they can be transferred to other places in the body, and they can also be used as like alternative energy sources and stuff like that, or they could be converted into lipids. And so when we talk about a ketogenic amino acid, this is something that can be converted into ketone bodies. So we call those ketogenic. And so these include the leucine, lysine, and lysine, which are strictly ketogenic, as well as some that are used for both, can be ketogenic and glucogenic. So glucogenic, they'd be used for glucose making, so sugar making. But this is a gross oversimplification. And what we're really talking about is something that's glucogenic is going to basically be able to be broken down into a component of that tricarboxylic acid cycle. So this oxaloacetate, kind of like this end product of the cycle, it can be used, it can be just cycled through again, merged with an acetyl-CoA to form citrate without the CoA part, and then kind of be transferred around. And basically the cycle, what it does, if you just keep cycling it, it's going to be producing these electron equivalents, these reducing equivalents, so like NADH and FADH2, which can then be used in the electron transport chain to make ATP. Or you can take out these intermediates and use them for other things. And so if you take out this oxaloacetate, you can use it to make glucose. And so if something makes a component of, if you can break down one of these amino acids into a component of the tricarboxylic acid cycle, then we can use it to make oxaloacetate, which you can use to make glucose. And so we call it glucogenic. But remember that it's not like you're going to, if you eat certain amino acids, you'll make a bunch of fat, and if you eat others, you'll make a bunch of sugar. It's really not that simple, and all these pathways are interconverted. And what the amino acids are actually going to be used for depends on the body's needs, as well as the cell type it's in and things like this. Finally, I've been talking about these like 20 main amino acids, but I should also mention that we talked about a couple of other ones. We have selenocysteine and pyrrolysine. These are the other two like genetically encoded amino acids. So basically these can be put into proteins already made, not post-translationally modified. And so these are selenocysteine, which our bodies have, as well as pyrrolysine, which some bacteria and archaea, so common of like bacteria-ish like things. Basically, these are a couple of weirdo amino acids that can get incorporated by tricking the ribosome into adding them instead of stopping. And much more on those in other posts. So you can find all those posts on my blog, the videos on my YouTube channel. And remember that although these amino acids are really cool individually, and we can learn about them individually, and some of them also have roles outside of proteins, things like serving as precursors for hormones. But really, their beauty really comes together when they're linked up into proteins. And when they're linked up into proteins, you have that they do so through merging their amino and their carboxylic acid groups. So they're no longer amino acids. The leftovers are going to be the residues, and the important parts is you have the backbone, which is going to be able to form those backbone interactions to give you the secondary structure, things like helices and sheets and strands. as well as the unique part, the R chain, the R group, the side chain, that thing we've been talking about and comparing these different side chains. And the side chains are going to be sticking out, and the protein is going to fold up in a way that best accommodates each of them. When it folds up in that way, you're going to have these residues in different positions. You're going to have these side chains sticking out in different places. And therefore, those places on the protein are going to have the ability, they're being like endowed with the powers of that amino acid side chain. And this allows you to have things like binding sites, as well as things like enzymes, like have active sites, places where reactions can happen. So really cool stuff that can happen thanks to all of these different amino acids. So hope that helped you appreciate them all.